Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about muscle. In terms of introduction, contractivity is a fundamental property of protoplasm, which is living substance, and it may differ in varying degrees. But in certain cells, muscle cells, they have a highly developed ability to convert chemical energy into mechanical work. Locomotion of multicellular animals, beating of the hearts, or movement of internal organs depend upon muscles of different types. And here you can see the cardiac muscle, smooth muscle, and skeletal muscle. So that's what we want to do today, and our objective is to identify smooth muscle, skeletal muscle, cardiac muscle in routine histologic preparation, and to examine the morphologic basis for different functions of these different types of muscle, and to distinguish between the modes of excitation of these muscle types. If this information is useful to you, please share it with your colleagues and your fellow students. Thank you. So there's four basic types of tissue: epithelium, connective tissue, nervous tissue, and today we're going to talk about muscle. Now, the function of muscle, in two words, is to generate contractile force. It contracts, and when it does, it pulls on tendons and ligaments that allows us to move. Now, muscles are different from other cells in that they have a high concentration of contractile elements, actin and myosin. Most all cell has that, but not at the high density that you have in the different muscle cells. And the actin and myosin could be arranged diffusely, like in smooth muscle, or they could be arranged in sarcomeres like in striated muscle, cardiac muscle, skeletal muscle, for more rigid contraction that we can see. Smooth muscle, and here we see striated muscle. It happens to be a cardiac muscle. So of the three types, you got skeletal muscle, very large cells, multinucleated, and the nucleus is in the periphery of the cell, as we see there. And then we have cardiac muscle, which is branched, as we can see there. A smaller cell, here you can see where it could branch. And then smooth muscle, kind of like a cigar, long cell, single nucleus. The cardiac can be one or two, but it's usually one. But here we see smooth muscle. And then there is another type of cell, the myoepithelium, that sends out like the legs of a spider around a gland to squeeze it. Cause milk ejection in the case of the mammary gland. Also, it causes dilation of your iris in the case of the eye. And here we can see the dilator muscle right in through there, myoepithelium, another type of muscle cell. But if we look at the three major types, skeletal muscle, we can see it in the tongue. And so here we can see the cells, the nuclei on the periphery of these large cells, multinucleated cell, and these are some capillaries inside there. And muscle cell is striated. You can see the striations in through there. Nuclei on the periphery, as we see there, with a high density of actin and myosin in the middle of the cell. Cardiac is branched. You can see where cells can branch. That increase the synchronization of the contraction to occur. You can see here where some cells are pulled apart. It usually has one nucleus in the center, not on the periphery. And where cells attached, this has been pulled apart, but you can see where it was attached, you have these intercalated discs. So you have additional striation. So this is your typical striation that you see in striated muscle, but this is where cells attach end to end in the cardiac muscle. Here you can see the intercalated disc. You can see the A and I band, and there's a nucleus there. And here we see mitochondria, very long mitochondria located in these cells. The other cell types are the smooth muscle cells, and a fusiform type cell that you see, kind of like a cigar, and you see the pink cytoplasm. It's usually a little pinker than what you see in connective tissue that you see. And so whenever the cell contracts, it kind of squeezes the nucleus and may even make it corkscrew, characteristic of smooth muscle. fusiform cells. Here we can see it in a ureter. These are bundles of smooth muscle cells. So there'll be several cells cross section. Sometimes they're longitudinal range, sometimes they're circular range. You can see the difference there. Here's the ductus deferens, more smooth muscle. So here we can see the individual cells there, cross sections of individual cells. Also in this artery, we can see smooth muscle in the wall. So of the three types, you got skeletal muscle, multinucleated, large cell, striated. It is voluntary. We can control it. I'm controlling it right now when I'm speaking to you. The cardiac muscle, involuntary. I can't tell it to speed up. I can think of something to make it speed up. Think of a bear, it may speed up, but I can't make it do it directly. And it's got usually one cell, sometimes two nuclei, one or two nuclei, and it's branched, so it can connect one another. And then you have the smooth muscle. It's involuntary. It covers sphincters and things, the paracelsus in the GI tract. One nucleus, non-striated. These two are striated. This one is not striated. There is connective tissue that surrounds muscle. And the outer connective tissue around the whole gizmo on the outside, which also has a lot of fat cells in it and fibroblasts in through there, is the epimysium. Mesium is muscle, epi is near or at the outside. And then we have perimysium, which is fairly large bundles of individual cells, and then you have endomysium, which is around individual cells. So the capillaries we were seeing before are actually in the endomysium that we can see. But the larger vessels would be in the perimysium, and of course the largest would be in the epimysium. And so here we can see those again. So it's epimysium on the outside. Here we can see some fat in through there. Perimysium would be major divisions inside there. And the endomysium will be what's around individual fibers. So if we look at the tongue, there's a little piece of tongue right in that area. And you can see the nuclei of the skeletal muscle cells. You can see a fascicle. So this is a fascicle which will be surrounded by a perimysium. And then maybe not every time you have a perimysium around it. Here we see the connective tissue of the perimysium around this one, right in through there. And then we see these individual muscle cells, and indeed they are striated as well. Striations, you can see very nicely nuclei on the periphery of these cells. Now, it's in the endomysium, the connective tissue thereof, that allows one cell to be attached to another cell. So the cells themselves are not attached to one another. They're connected by the connective tissue of the endomysium around them. Unlike cardiac muscle cells, where the cells are actually attached to one another. But this allows you to have multiple cells along the length of a muscle, so you can have a long muscle long enough that you need. And so the perimysium has larger blood vessels running in through there, but it's the endomysium where you have the capillaries that you see there that are actually touching the individual muscle cells. And so these capillaries here that we see there in this tongue, so this would be an individual muscle fiber there, there's another fiber there, another fiber, a muscle fiber, muscle cells, the same thing. And here we can see the endothelial cells of the capillaries in the endomysium. Endomysium, we can see here and here. This is individual cell nuclei on the periphery. This is a cross-section of those. A longitudinal section would yield the A and I band. The A band is the anisotrophic. In other words, it does alter the birefringence. And then the I band is light band, isotrophic. It does not alter. In other words, it stays the same. This does not stay the same, so therefore it changes it. And so the A band is dark, which has the myosin in it, and the I band is light, which has the actin in it. And so here we can see the A, the dark band, and the I, the light band that we can see there. And here we can see polarized light. This is a website that showed this. You can see the I band and the A band located in through there. So if we see the A band, it's a myosin, I band, actin. In between there, we have a Z line. So a sarcomere is from Z line to Z line. that we can see there. And you remember, it's the endomysium that connects one cell to another cell, even though here we see the nuclei of the cells on the periphery. But if you look at the sarcomere, which is the unit of contraction for rapid contraction and rapid control, from Z line to Z line is one sarcomere. That's one sarcomere, about 2.5 micrometers in thickness. The Z disc that we see here and here, the thick filament, which is a myosin, and that's what's in the dark band or the A band, and the thin filament is in the light band, but also there's some crossover into the dark band themselves. The myosin stays put in the contraction. It's the actin that moves in relation to sliding filament theory. So the sarcomere is the structural and functional unit of muscle. This is a nucleus up there, and we see a sarcomere from there to there. The I band is in the middle. The A band is in the middle of the sarcomere, and the sarcomere is composed of two halves of the I band. So I band is not, the complete I band is not in a sarcomere, is two halves in it with the complete A band in there. You also have the M band and the H band that's located. H band is where the actin does not overlap with the myosin. So if we look at the filaments, we have thin filaments, which is actin, and the thick filaments with their heads connecting sites there, that would be the myosin. Now, the thin filament, actin, is associated with proteins. When it's associated with proteins, then it causes actin to do things. If it's associated with filament, it makes a gel. But in the case of it being interactive with tropomyosin and alpha-actinin, which is what's in the Z line, then they become arranged linearly, as you see. And then how they would interact with the myosin in the middle. So there's bundles of contractile filaments that you see there. They started with the tropomyosin interacting with actin, making them a more linear structure in the Z line or Z disc being developed. And so basically what we see there, this is what we see there. This is a Z disc here, and that would be the alpha-actinin, and you would have the myosin, actin on either side. And so here we see the monomers of actin, and then you got tropomyosin, and also have a troponin. Troponin is the proteins apparatus that facilitates the precise control of sarcomere contraction. And they have three components. They have a T component that covers the binding site, or it actually binds to the tropomyosin, I should say. Then it has a calcium binding and inhibition site that we see there. So that's note the uniform spacing of the troponin. Uniform spacing, very important. And so here we can see the actin and the tropomyosin and the three different components of the troponin complex. And now in muscle, there's 13 isoforms in myosin, 128 isoforms of tropomyosin, of troponin. We don't know exactly why they're there. They're just there. And here we see muscle. This is a nice body whirl thing showing you the muscle there. The myosin heads that we see and the troponin that we see there. And remember, you got the DNA giving rise to RNA, and you got the splicing, and then you have a little bit of protein produced, and this can be modification during the process to make the isoform. You can see here we have the exons and introns from the genes that go there, and then finally is duplicated. Introns are cut out, and you have the messenger RNA. Now, in terms of footprints of evolution, one is fossils, and we'll see one here in a minute, and another one is the biological molecules, and also the anatomy and physiology. So if we look at the anatomy next, you can see that based on these animals, what characteristics is similar to these animals. What makes a mammal? All these are mammals, and here's one of them. They don't have feathers, they don't have scales, and the mammary gland, that's their name, mammals, and they have a special ear bone. And here we can see a fossil that we can see right here. This is early reptile, and this is a mammal-like reptiles that scientists hypothesized for a long, long time before they finally found one. So in us mammals, modern mammals, we have one, the dentary bone is right here, right there. In a snake, they have two additional bones. And these two additional bones left the jaw and became our inner ear bones. And that's why special inner ear bones, what makes a mammal different from a snake. Okay, finally scientists found one. Here you see a penny. It's not a very big animal, but they found this mammal-like reptile, 125 million year old fossil with inner ear bones, intermediate, still attached to the jaw between a reptile. Now, in early embryologic stage of modern mammals, that's us. When we were embryos, the middle ear was still attached to the jaw. So that's why scientists knew about these, because they saw them in the embryo first. It's in every embryo, if you're a mammal. That's where it is. So fossils is one footstep. Another one is comparative anatomy, as we talked about. It's one thing that you can do. And here we can see, you can learn a lot about humans by looking at animals. And here we can see this is man and the man-like apes. So we actually part of an ape, right? We're more similar to apes than we are to monkeys. Also, biological molecules. If you look at cytochrome C, which is a protein that is in the mitochondria. If you look at it in rice and tuna, it's very similar. How can that be? How can that be that they're similar? Did the tuna eat the rice? How did it get? And so here, somewhere the tuna would be over here, but there's rice way over there. So 100 million years ago, we were on the same level. So we're all derived from the same type thing. That would explain why they are similar. And how does mutations occur? How does evolution occur? One is by point mutation, where you have a change in the function, where you change, and you may change the function of it to do something different. Maybe a duplication. Like one of these would be myosin, and would be hemoglobin and myoglobin. Myoglobin is 1 unit, as opposed to hemoglobin is 4, and so you can have duplication and make hemoglobin from that. Exon shuffling and alternate splicing. These are different ways of which proteins can evolve. And here we see the exon shuffling. So you can make different proteins. You can make kind of mosaic proteins from these mosaic because you can combine A, B, or C, A, B, and X, A, B, and Y, different combinations that you can make these mosaic proteins that you can make. And so here we can see different exons being involved and different combinations. Note that the order is the same. What's different is which one of these are pulled in. So they all have green, and they're all in blue if they have blue. This one doesn't have blue, you can see. But you make these combinations off of the same gene. So this is isoforms produced by alternate pathways of making these protein isoforms. So, as I mentioned, 13 isoforms for myosin, 128 isoforms for troponin. Why we need it, we don't know, but we're glad that we have it. So if we look at the structure of a muscle cell, the myofiber is the muscle cell, so the fiber is the cell, multinucleated cell. The myofibrils are components that are inside the muscle cell. And the sarcomere is composed of different things. From Z-line to Z-line is one sarcomere. It has the I band and the A band. It has the H band where you don't have overlap to occur. And you can see that here. So this would be where the H band would be right in through there. And this would be the A band, and then this would be the I band that we see there, with the myosin staying put and the actin migrating. So here we can see individual cells that are said that put together with endomysium to hold cells together. So if we look at the fascicle of cells, we can see though this is the individual cell, individual fiber right here. So this is the same as that. If you cut it open, you would have these myofibrils. Myofibrils are the contractional units inside there, which has the sarcomeres. They house the sarcomeres. And then if you look at one sarcomere from Z line to Z disc to Z disc, and if you cut through here, you can see different things. Just actin in the I band, just myosin in the H band, or right in the middle you have the complex that holds the myosin. But if you cut right through there, very interesting, you have both actin and myosin that we see. So here's actin and myosin, and it's the myosin heads that cross-link between the myosin and the actin. And remember, this organization all came back from the actin interacting with the tropomyosin, which made it a linear structure that facilitated this to occur. So skeletal muscle, we see a sarcomere from Z disc to Z disc, the A band, the I band, H band. And here we can see the section through actin, 6 actins with 1 myosin that we see very uniform that we can see there. So this is a cross section right through there that would yield that type of view. How about wallpaper? Smooth muscle wallpaper, wouldn't that be something? See, 6 is a magical number in nature. 1, 2, 3, 4, 5, 6. How many sides do you have of a bee cell, of a honeybee's the chamber? How many sides? You got 6. 6 is a magic number in biology. And here we can see what happens. So the myosin stays put, which is red here, and the actin moves. So the Z line, Z disc, get closer together or further apart. That's what it is. And so here you can see in a contracted state, you can hardly see any H band. H band can be seen in normal, but H band is very thick where there's no overlap if the muscle is stretched. And so, again, the myosin stay put. It's the actin that changes, and that also changes the location of the Z disc. Now remember the intermediate filaments in epithelium. Remember they were structural components in epithelium, the hemodesmosomes as well as desmosomes. In muscle, it's intermediate filaments that hold the myofibrils organized. Without that, you might not have as precise contraction to occur. These intermediate filaments that does that. So they hold the myofibrils in connection with the muscle fiber or the muscle cell. Now, if we look at contraction of the sarcomere, which is a functional unit and a structural unit of muscle, we have the different components. The T part attaches to the tropomyosin. So you have attached to the tropomyosin. C part binds calcium. And the I part inhibits the actin binding interaction site. And so those are the 3 parts that we have. The calcium binds here. This part binds to the tropomyosin, and this part is inhibiting the binding to occur. And this is a sliding filament theory of contraction, that the myosin stays put and the actin slides. And so there's basically 5 steps to it. You got the troponin C, the C part binds calcium, as it does, and it causes a transformational change in the troponin as well as the tropomyosin. And the tropomyosin moves out of the way, so now the actin site is exposed. So myosin binds actin and releases inorganic phosphate. In doing so, it binds and movement of the head. So the head moves. When the head moves, that is actually the motor or the power stroke that does that. And then, to recock it so it can move again, the ATP binds to the myosin head to change that to ADP and inorganic phosphate, and that cocks it for the next one. So it's cocked here, it's already rotated there, and to cock this again, you need to have ATP to do that. So those are basically 5 steps associated with it. And so here we can see it again. If you note these 2 red balls is attached here This is the myosin binding site of the actin. And so you have it attached. ATP causes this thing to cock it. Whenever you have that, it cocks it. And then it binds to the next site, and then it slips it further. See, from here to there, it cocks it, and then it binds to that site, and now it slipped further, and that's how it moves things along. So calcium regulation is involved. You have a transverse tubule. So if this is a cell that you have, this is a cell membrane. Invagination of the cell membrane is a transverse tubule. So you have modification of the cell membrane by the membrane projecting into the cell. You normally don't have that in a cell. And it touches the sarcoplasmic reticulum on either side. So the transverse tubule in blue, sarcoplasmic reticulum in here, makes so there's two sarcoplasmic reticulum, one on either side. And this is a smoothie of the muscle cell, and that holds calcium, and it sequesters calcium after it has occurred. And so here we see a triad, two sarcoplasmic reticulum, and the transverse tubule that go there. And so sarcoplasmic reticulum responds to the action potential coming down deep through the cell. So the action potential is on the surface, migrates down, and it stimulates sarcoplasmic reticulum to release its calcium. When it releases its calcium, then so this is the cytosol here. This is inside the sarcoplasmic reticulum. The calcium goes out and binds to the troponin, as we've seen before. And this is due to the action potential going from the cell surface down deep within the cell itself. And so, again, we can see the different components. You get the nerve working with the motor end plate of a muscle cell, and that changes the action potential of the plasma membrane, and that then goes down deep into the cell itself, and the calcium stores, which are in the sarcoplasmic reticulum on either side there, is released, and that binds to the troponin, which then frees the actin binding site to myosin. Myosin binds, and then you have a power stroke, and then you have to have more ATP for the cocking of the myosin head again. So here we see the motor end plate. So we got a nerve here interact with multiple muscle fibers. You can even see the striations in the muscle fibers. So the motor end plate, it has this kind of infolded part. You got the synaptic cleft in through there, and junctional folds. So these junctional folds increase the surface area of the muscle cell so that you can have more exchange with the nerve. And so the nerve's coming down here, the motor end plate, in through there. One nerve may innervate more than one muscle cell. In fact, it does. And here we can see the motor end plates at the end of these nerve fibers. So these are motor end plates in through here, and here, and it nerve innervates more than one cell to contract multiple cells along at the same time. And you can see these folds, junctional folds, in through there in the motor end plate. So what happens is you get the action potential coming down the nerve, and whenever it gets there, it causes a calcium channel to open, exocytosis of the transmitter. Transmitter then goes and interacts with the muscle cell, and that changes the calcium, sodium influx, and in doing so, it causes calcium release from the sarcoplasmic reticulum. So this is action potential moving down and then going inside the cell, interact with the sarcoplasmic reticulum to release calcium inside for contraction to occur. So here we see innervation of the muscle. This is muscle cell here. This is the nerve with the secretory transmitters in through there, and they'll be put into these junctional folds, which is amplification of the cells so that you can increase the surface area. There's a myofibril down there. So here we can see the synaptic cleft, acetylcholine receptors located right in through there to respond to the acetylcholine nerve interacting with more than one muscle cell. At the synaptic cleft, you have the vesicles right here. The synaptic vesicles go through here, interact with the surface there, as we talked about before. Here at the presynaptic site, you can see vesicles right underneath the membrane. They're just about to be released, and you can see where some are in the process of being released. So this is exocytosis caused by calcium influx. In addition to a motor unit, we also have sensory, and this is a muscle spindle. This is a muscle spindle. You can see a cross-section through there, and these are actual muscle cells inside there, which are in a capsule, and these are stretch receptors. You can see again, it's a longitudinal view of a similar type thing, a muscle spindle. The intercapsular fibers that we have, or we call interfusal fibers, they're located right there. Another one that we can see here in the tongue, you have muscle spindles in the tongue. So this capsulated structure right here is a muscle spindle. These are nerves that we can see. You can see the myelin sheath, the darker around the axon. And then these are fibroblast cells, and through there, these are capillaries in the muscle cells. And these are nuclei of the muscle cells themselves, but this is a muscle spindle. There's another muscle spindle here, and these are the interfusal fibers, or the muscle cell fibers inside these. These have, they're striated, they're really modified skeletal muscle cells. So there are different types of fibers. You have the red type, which is slow oxidative. You have the white type. And so where you're eating dark muscle on a chicken or white muscle, it has to do with the different types. There's intermediate ones too. Fast. If you were a distance runner, you'd have more red. If you were a sprinter, you would have more white fibers. And here we can see muscle. Wherever you go, there goes your muscles. And we see the muscles in there. And here we get to the cardiac. Cardiac, we have some valves inside there, but we have the major cardiac muscle that we see there. Cardiac muscles are branched, as we mentioned before, nucleus in the middle, and you can see cross-sections of those vary in size because you have different sizes of the ends of them for them occur. Well, they are attached at the Z-line by the intercalated disc, and that's a vascular adherence, macular adherence like a desmosome, and the gap junctions are usually on the lateral border. But the cells attach to one another from end to end, as you can see there. Intercalated disc is the attachment that we have there. So the attachment and the gap junction is usually on the lateral borders, as you see, but still this is all part of the intercalated disc that we see there. Intercalated disc is always on the Z-line at the end of a sarcomere that we can see. There it is right there. And so this is the sarcomere in cardiac muscle. Sarcomere, sarcoplasmic reticulum, I should say, is the smooth endoplasmic reticulum, and that's the one that's going to have the calcium regulation. You can also have glycogen stored in muscle as well. Now, in a cardiac muscle, you have a dyad, not a triad, because the sarcoplasmic reticulum is not on both sides. It's not continuous on the both sides. Both sides touch it, of course, but it's not a continuous line. So when you cut through it, it's more likely you just have one component of a sarcoplasmic reticulum, and that makes a dyad. Dyad is transverse tubular plus one end. In skeletal muscle, you had two sarcoplasmic reticulums. You have two sarcoplasmic reticulums here. They're just not along the sides so that you cut it in a triad every time. There's also these apical granules that are in muscle cells that have to do with some hormone secretion and regulation. You also have capillaries. This is a capillary in muscle, a nerve in the cardiac muscle cells, or lots of mitochondria in through there. There's a nucleus of a cell. There's a pericyte that we see there. And so here we see the cardiac muscle organization in terms for contractions. You have the SA node, and then there's some fibers that connect here, the AV node. AV node then sends Purkinje fibers down deep. Purkinje fibers go down to the apex and then go back up so the ventricles can contract. Purkinje fibers are electrical transmitting cells that are modified cardiac muscle cells. Here we see the internodal connection between the SA node and AV node, and then these are the bundles of His or the Purkinje fibers that we can see there. Here we see the Purkinje fibers. They look different. They're bigger than the regular cardiac muscle cells. These are modified cardiac muscle cells, not nerve cells, and they have a lot of glycogen in them. That makes them big, and that makes them light staining because the glycogen is usually removed during the processing of a tissue. But you can see myofibrils inside there. And you can see a lot of glycogen in there too. And there's a lot of junctions between there. Nixus junction here, which is a gap junction. So there's a lot of junctions there facilitate communication from cell to cell. So these are in the, these are Purkinje fibers that are in the heart. Here we see the heart pull back, that we can see the cardiac muscle located there. The other muscle that we see through there is smooth muscle, and that's what we're going to talk about next. The cell organization, the myofibril organization, intermediate filaments, and fusiform densities is what we're going to see in there. So smooth muscle, the gut has a lot of smooth muscle because it's in charge of paracelsus, the grinding, mixing of food that we see. So this big layer of wall of the intestine is smooth muscle. This pink thing here is smooth muscle, and this in through there is connective tissue. We can see it again, big bunch of smooth muscle in through there. Also, there's muscle in through there, and this is connective tissue that we see in through there. There's also muscle in blood vessels. You got smooth muscle. The muscles and blood vessels constrict the flow, is what they do. The smooth muscle does not move blood forward. It does move food forward in the digestive tract, but not so in blood vessels. The pressure is produced by the heart, by the cardiac muscle. And the blood vessels, the smooth muscle restricts the size so they can regulate the flow. Also, we have smooth muscle in the rectopili muscle. If you ever had chill bumps or cold bumps, then that's the erector pili muscle trying to pull the hair up to stay warmer, but meanwhile it indents the surface there, making the chill bumps. Smooth muscle, as we can see, individual cells have a basement membrane. In fact, if you use PAS staining, you can see the basement membrane around the cell. So in that case, smooth muscle is like epithelium. It has a basement membrane. Also, smooth muscle, remember before we said it had an actin, which are these small filaments, and then the myosin. The difference is organization. It's not organized in a sarcomere. But we have that, so there's a myosin and there's an actin that's inside there. And on the surface, it has these apical caveoli on the surface in through there, and in there you engage calmodulin to bind calcium for different kinases to be involved to activate the cell to contract. Not sure exactly how it works. It's different from the skeletal muscle contraction, and it's thought to use the apical invaginations in the process. So these are intracellular caveoli that are on the surface there. And so basically you have phosphorylation of the actin and myosin somehow to get binded to occur. Now, one thing I do want you to remember is the filaments that are in there. There are different types of filaments. The blue ones are the intermediate filaments, and they don't change. They don't contract. But the actin and myosin change. They change. And so you can see there's myosin, which got shorter here and got shorter here, and then the actin filaments that are there as well. And so what happens is the actin and myosin changes its shape. Intermediate filaments stay the same. In doing so, it squeezes a cell. So the cell gets from a cigar to more like a balloon, like that in the process, and it squeezes the nucleus and maybe make it corkscrew. And where these filaments attach to one another, sometimes on the cell surface, sometimes intermediate, that makes these structures here that we see, these dark structures that we see there, some on the plasma membrane, some inside the cell. These are the dense bodies or the fusiform densities that characteristic of smooth muscle. Smooth muscle also has gap junctions to communicate between the cells. So in terms of regeneration, cardiac muscle, none. You get a heart attack, you don't replace the muscle cells. Skeletal muscle, some, usually it's the size. You start lifting weights, your muscles are bigger, they increase in size and not number. Smooth muscle, lots of smooth muscle data. Here we see comparison of cross-section of the filaments associated with skeletal muscle, this is striated muscle, I should say, and smooth muscle. You see both of them have actin and myosin. The difference is the organization of the two. It's more fusiform, more dispersed, diffuse, I should say, in the case of smooth muscle, as opposed to very organized sarcomeres in the case of striated muscle. So in summary, you have different shapes. You have nuclei on the periphery, nuclei that are in the center. You have multinucleated, you have double or single nuclei in cells. You have the sarcomeres facilitating contraction, control with a troponin and a tropomyosin, contraction of smooth muscle, squeezing of the nucleus, the fusiform densities that you see there on the cell surface as well as inside there is where the filaments attach, and you have the nerves telling the cells to get going. And here we see muscle associated with our play that we have. And there's several books that facilitated us to be able to get these illustrations. We want to thank those books. Also, thank Body Wearers for a couple of the pictures that we have there. Excellent presentations of muscle cells throughout there. Next time, we're going to talk about the peripheral nervous system. Either that or we're going to go to Paris. And there you see the Eiffel Tower in Paris. We also saw castles that were there. And so we see castles. If this was useful to you, please share it with your neighbor, with your colleagues, with your college overseas. Thank you.